what I'm going to be talking about in the next few minutes is sort of coordinating biochemistry and cell biology, because, of course, what the biochemistry that we study actually occurs inside of cells, or at least the stuff that's particularly interesting. And the cell biology I'll talk about here has to do with export of RNAs from the nucleus into the cytoplasm. And I will actually, again, break the talk into two sections. One is, I'll give you a general feeling for how RNAs in general are taken from one cell compartment to the next in a specific manner. And then I will also say a few words about proofreading of tRNAs and the processing of tRNAs. Now, general features of RNA export are that it's primarily a directional event. It will go from the RNAs are made in the nucleus, oftentimes function in the cytoplasm usually, and this transfer from one side to the other of the nuclear envelope requires energy. And there are two different sources of energy depending on the RNA. GTP is often hydrolyzed to GDP in conjunction with the protein that shuttles back and forth, which is called RAN, and I'll get into that in a minute. And then there's also ATP hydrolysis that is involved in primarily in messenger RNA export. RNA export is mediated by specific carriers called export receptors. And the receptors can either bind the RNA directly or by way of an adapter. And I'll give examples of this in a minute. Now, when we think about the nucleus of a cell, oftentimes you'll see that drawn in a schematic diagram as if it's a little balloon that is just a round circle. And this stuff happens inside, and magically it gets into the outside. Sometimes it's drawn that way. Other times it's drawn as a line, the nuclear envelope, as a line with a bunch of holes in it, as if there's just a screen or something. Things could go right through. Neither one is correct. What happens in in cells is that there is an envelope called the nuclear envelope. It's a double membrane that is what we often see as the balloon. And that envelope is punctuated by structures, very complicated structures, huge. They're much bigger than the ribosome. Ribosomes have to go through these are called nuclear pore complexes. Ribosomes go through those nuclear pore complexes from the nucleus where they're assembled into the cytoplasm. I've just shown a blow-up of the nuclear pore complex here that I just got off the web, which shows on the nuclear side there's something called the nuclear cage, and on the cytoplasmic side there are fibrils that go into the cytoplasm. And transport between the nucleus and cytoplasm goes through these nuclear pore complexes, and it's directional. Some molecules go into the nucleus, others go out of the nucleus. I'm not going to talk about nuclear import today for lack of time, but we'll just talk about the export from the nuclear side into the cytoplasmic side. RNA molecules, well, transport between the nucleus and the cytoplasm is size limited. Small molecules can diffuse through these nuclear pore complexes, but when you get up to about 20 or 30,000 molecular weight, you have to go with a carrier, and the carrier is depicted here just as in a model. So the RNA cargo can bind to the carrier or associate somehow with the carrier, and it's really the carrier that mediates the transport across the nuclear envelope, and in the cytoplasm the RNA is released. Now, I mentioned the hydrolysis of GTP as being an important feature in export. The GTP actually gives directionality to the transport, and an example is here. There's a protein that cycles between the nucleus and cytoplasm called RAN, R-A-N. It's a small RAS-like protein. It binds GTP, and when it binds GTP, it changes its shape and it can interact with an export receptor, or exportin. And association of the RAN GTP with the exportin allows the exportin to bind its cargo. Okay? And when that happens, then this ternary complex, or actually in this case it would be quaternary if you consider the GTP, this is the complex that actually is exported to the cytoplasm. And in the cytoplasm, there are proteins that are primarily cytoplasmic that activate an enzymatic activity of RAN. And the activity that's activated is a GTPase. So in the cytoplasm, these proteins, RAN binding protein and a RAN GTPase activating protein, stimulate the RAN to hydrolyze the GTP to GDP. And as you can see schematically, the shape of the RAN changes. And when it changes, it no longer has an affinity for the exportin, and so it releases the exportin, which is now free to release the cargo in the cytoplasm. And so what we have is a directionality of taking something from the inside to the outside, okay? And the GTP hydrolysis accomplishes that. And now all the components have to be brought back into the nucleus. Okay, so a word about these exportins. Probably a dozen years ago now, several groups, primarily Dirk Gerlich in Germany, but other groups as well, isolated a number of export receptors on the basis of their ability to bind RAN GTP. And they came up with about 12 to 15 different export receptors, different proteins that would bind RAN GTP. And it turns out that each of these has a specific cargo that it likes to interact with. One of the first ones that was isolated is called CRIM1, or exportin-1. And this is a protein, an export receptor that is specific for proteins. It'll bind proteins that have specific signals on them to allow their export. So CRIM1, in association with RAN GTP, will bind a protein and take that protein into the cytoplasm. And then do the GTP hydrolysis as we discussed. Some other export receptors are specific for cargoes. And so there's one called exportin T, which Ian Matai and Elisa Izarralde studied several years ago. Exportin T binds transfer RNAs directly and carries those transfer RNAs into the cytoplasm. It doesn't carry out protein. One that we've studied is exportin 5, and I'll talk about that in a minute. Exportin 5 binds structured RNAs in general. It's one highly structured RNA is the pre-microRNA that you heard about 2 days ago in a couple of talks. And exportin 5, again, in the presence of RAN GTP, will take these precursors into the cytoplasm. Sometimes the cargo will not bind directly to an export receptor, but has to use a protein as an adapter. Okay? Not all RNAs will bind to export receptors. And in this case, we call it an adapter, and I've just drawn it as a little pentagon here. So it's the adapter that would bind to an export receptor, oftentimes CRIM1, actually, the adapter will bind to the CRIM1, and the adapter itself binds to an RNA. Okay, so now this is just making a bit of a more complicated structure that has to be. Okay, so one of the earliest examples of an adapter that carries an RNA into the cytoplasm was processed RNA from HIV virus, which actually is a messenger RNA, and there's a protein that will bind to the HIV messenger called Rev protein, and that Rev protein will interact with CRIM1, the protein adapter, and so the RAN-GTP CRIM1 complex is responsible for taking this particular form of the HIV message into the cytoplasm. Okay, so this is a case where a messenger RNA is brought into the cytoplasm by way of interacting with an adapter, the Rev protein. But in fact, most messenger RNAs don't use this pathway. Most messenger RNAs are exported using another pathway that is actually independent of RAN-GTP. And this is work initially out of Israeli's lab. TAP and P15 are two additional proteins that will interact with messenger RNA and carry that messenger RNA through the nuclear pore complex to the cytoplasm. And there are more than one way of getting a messenger out through the adapter pathway that I talked about, as well as through the TAP pathway. But when a messenger RNA is exported to the cytoplasm, it brings with it a lot of proteins. In the process of going through the nuclear pore complex, a lot of proteins are able to stay on the messenger, but a number of them are removed as well. And this is a schematic view from Ed Hertz's lab that shows that here we have a messenger RNA that's being spliced in the nucleus, and during splicing events, as you've heard, some proteins are deposited at the exon-exon junction, the new one that's formed during splicing, and this is called the EJC, which Lynn McQuat studied and discovered and showed very nicely that this plays a very important role, which I guess you'll be talking about this afternoon. Okay. So the exon junction complex stays on the message as it goes through into the cytoplasm and plays a role in what Lynn will be talking about, the nonsense-mediated messenger decay. Here's TAP and P15 that go on to the whole complex. They come through the nuclear pore complex, and it's thought that they're responsible for actually taking it through this pore. There's a very important other proteins that are involved on the cytoplasmic side of the complex called DBP5 and GLI1. DBP5 is an ATPase, and the ATPase activity is responsible for release of the messenger once it comes into the cytoplasm. It's released from a lot of the other proteins that brought it along. So something has to allow the messenger to be released and function in the cytoplasm, and it's DBP5, which is activated by GLI1. So it's a very complicated series of reactions, and that's all I want to get across right now, that these things don't just diffuse out, they are brought out in a unidirectional manner, and that uses the ATP hydrolysis to accomplish that. Okay? So that's just so I can sort of cover the waterfront on RNA export. Now I'm going to talk about some work that we've done, the export of pre-microRNAs and transfer RNAs, and then I'll go on and say a little bit about how this can be coupled with proofreading of these molecules as well. The system we use is the Xenopus laevis frog, and the assay, which I'll be using over and over again, is shown schematically here on the right. We can inject RNAs into the nucleus of a frog oocyte, incubate the oocyte for a while, and after some time, depending on the kinetics we want to study, we can dissect the oocyte into a nucleus and a cytoplasm and fractionate the RNAs that remain in one cell compartment or the other. We always put into our mixture of experimental RNAs a molecule of U1 RNA. These are small nuclear RNAs. U1 normally goes into the cytoplasm, and this is a mutant form that stays in the cytoplasm. And U3 small nuclear RNA remains in the nucleus. And the reason we put these into all of our experiments is that it allows us to know whether we've hit the nucleus in the injection to begin with the needle, whether the cells are dead, and do these we know how these RNAs should behave. And so we always check our injections to make sure that those end up in the appropriate place. And then we can see where the experimental RNAs are. You've already heard about this. MicroRNAs are made in the nucleus as precursors, which are exported. And this is the event that we're interested in studying right now, the export of the premier from the nucleus into the cytoplasm. This is an actual mixture of RNAs that we injected in here, U3, U1, and a mixture of precursors of small nuclear RNAs. And as a function of time, this is in hours, you can see that what we put into the nucleus, the premiers are very rapidly exported to the cytoplasm, and very little stays in the nucleus, and then the controls worked as expected. It takes a while for the U1 RNA to get out to the cytoplasm here.
Are those hours? Or?
Those are hours, yeah. If we deplete the oocyte of RAN GTP, and we can do that by injecting a dominant negative mutant of RAN, so it uses up all the RAN GTP. You can see that the U1 does not get into the cytoplasm, and also the premier RNAs also don't get into the cytoplasm effectively. So what this shows is that these export of these RNAs is RAN GTP dependent. You can also see that it's very quick, very fast. So now we can ask questions about what is it that is actually carrying these precursors into the cytoplasm. I already gave you a hint about this in an earlier slide. This is just, again, a repeat of the same kind of injection experiment. The controls up here. Here we used a mixture of a tRNA, Med-I, and Premier 31. That's just a precursor of microRNA 31. And both of these RNAs get into the cytoplasm very rapidly. Now we can add a large amount of competitor. Since these are molecules that are being exported by a carrier, there's only a fixed amount of carrier in the cell. So if we put in an excess of unlabeled competitor, we should be able to block the export of the radioactive RNA. And in fact, that's what we can see. Here we've put in the competitor of the cold premier and a lot of the cold transfer RNA, and you can see it has no effect on the U1 export. In fact, it seems to be a little bit faster. But these two molecules are primarily now in the nucleus rather than in the cytoplasm. So we're competing for the carrier that would bring them out. And since we have now, thanks to a number of other labs, we have at our disposal cloned versions of all these export receptors. We can add back individual export receptors and see which ones bring these molecules into the cytoplasm. And you can see if we put in exportin 5, we now are able to see the pre-mir 31 appear in the cytoplasm. So that says that what had been saturated by the competitor is exportin 5, the endogenous exportin 5. And this is not at all a surprise because it's been known for several years. Exportin T now rescues the export of the tRNA, but it does not rescue the export of the microRNA. Okay? So that shows the specificity of these two different export receptors. And just as a control, we also put in the CRIM1, the carrier that brings proteins into the cytoplasm, and that has no effect on either of these, showing that export of these two RNAs is not mediated by CRIM1. The interaction between the premier and exportin 5 is direct. We have, as I said, we have recombinant exportin 5, and we can mix that with the premier in the presence of ran GTP and do a gel shift where the complex is retarded on a gel because it's larger. The radioactivity is in the premier 31, and when it binds to exportin 5, it migrates more slowly on the gel, and you can see that's the case, and it binds directly. We didn't add anything other than the RNA and the exportin 5. So it's a direct interaction. So just to summarize this, exportin 5 mediates nuclear export of premier RNAs, binds directly to the to the RNA. Exportin T is specific for tRNA, and both require ran GTP. Okay, now I'm going to say something about tRNA export and monitoring of tRNA integrity, or the proofreading, basically. As you've heard several times, especially today, tRNAs are made as precursors. With an intron, there's a 5', often 5' sequence over here. There's a 3' trailer sequence, and all of these events have to be, all these extra nucleotides have to be removed in order to generate a functional tRNA. Generally, the 5' end is removed first by RNase P. The 3' end is removed again by a specific nuclease, and then the CCA end is matured. And in vertebrates, the intron is removed here. Okay? And all of this happens, at least these processes happen in the nucleus. And we just made an artificial tRNA with the 5' trailer. We were interested in whether the ends are necessary for export, mature ends. So if we put this RNA into the nucleus of an oocyte now and look at export from the nucleus into the cytoplasm, this is what we injected, and you can see none of that gets out into the cytoplasm. It all gets processed down here, and when we look at what that processing is, it's removal of the 5' extra nucleotides here. And this RNA, which was processed, now is rapidly exported to the cytoplasm, saying that you need to have removal of that end, of the extra nucleotides of the 5' end. So what about the 3' end? Well, in order to investigate that, we put in a truncated version that did not have a CCA, it was just truncated with nothing after the acceptor stem in the double-stranded region. And that material gets cleaved by RNase P, so it loses its 5' extra nucleotides. But this is very, very slow to get out into the cytoplasm. Okay. And in fact, it essentially doesn't get out. A little bit of the RNA that we put in does get elongated, probably because we didn't remove this the 3' tail completely, and so that just accounts for the little bit that got out. But what we conclude from this is that both ends have to be intact before the RNA gets exported. So how does a cell know that the ends are mature? Well, and that the RNA is ready to go into the cytoplasm. Well, one way you can imagine is that you can say, well, perhaps the tRNA has to be capable of being amino acylated, because after all, that's what's going to happen once it gets into the cytoplasm. Okay? So we asked whether, in fact, well, what that would predict is that there would be some kind of amino acylation of the tRNA in the nucleus to allow for its monitoring. And so we asked whether tRNAs can be amino acylated in the nucleus, and this is the kind of experiment we did on that. To do that, this is looking at tRNA met-I, a little squeezed together here. When we run the RNA on a gel, just isolated from what we've done is we inject this tRNA into the nucleus, and then after waiting a while, the RNA is rapidly exported, and we isolate the tRNA, and we ask whether that has an amino acid on it, and as you would expect, it does, because it's being amino acylated very efficiently. And our assay for amino acylation is to run this RNA on acid urea gel. It just is a gel that Tom Rajbandari developed, which is able to separate amino acylated from non-amino acylated tRNAs. And they separate on the basis of gel mobility. And excuse me. So the amino acylated tRNA is a little bit slower than the uncharged tRNA, and we can see that difference just by taking the amino acylated RNA and allowing it to be deacylated mild high pH incubation. And a second, excuse me. So, so this is the assay. Is there an amino acid on that RNA? And this is not surprising that there is. We can keep the tRNA in the nucleus. Remember I said we injected the RNA into the nucleus and then took what was in the cytoplasm. We can keep it in the nucleus by removing the RAN-GTP and that, from the system, and as we did before, that blocks export. And now we can see all the tRNA that we injected is in the nucleus. And now we ask, is that RNA amino acylated? And yes, indeed, it is amino acylated, even though it never got out into the cytoplasm. So that shows us that amino acylation does occur in the nucleus. So the question is then, what is the function of that nuclear amino acylation? And we postulated that it might facilitate nuclear export. That's the whole idea here. And to block amino acylation specifically in the of the tRNA, we put in an inhibitor of amino acylation, and this is a compound that's very similar to the activated tyrosine that is put onto the 3' ends of or used to charge tRNAs. as Dino was talking about earlier, tyrosine AMP is the intermediate that is normally used. This is a compound that is isosteric with tyrosine AMP, but it has an SN bond instead of a PO bond, and it specifically inhibits tyrosyl synthetase as a competitive inhibitor. And we put tyrosyl tRNA into the nucleus of these oocytes and asked how this compound affects export. And as you can see, it slows down the export of this tRNA. I don't have it here in this slide, but it has no effect on the export of other tRNAs. Okay, so it's specific for that particular tRNA. And we've done it with other sulfamylyl or AMS derivatives for other amino acids. And again, it's always specific for the tRNA that we're targeting. And so we conclude that charging is important for efficient export. So just to conclude what we've shown on the tRNAs here, ends have to be mature. Nuclear aminoacylation accelerates the formation of complexes for export, and therefore there's some kind of functional proofreading. And now that tells us about the 3' and 5' ends of these RNAs, and what actually is exported in the oocyte system and vertebrates in general is the mature tRNA, okay, which we have down here. We felt for a long time that this was the end of the story, but it isn't. And the reason it turns out that it is not the complete story is, as Glauco was telling you, intron removal in yeast is different from intron removal in vertebrates. And what happens in yeast is that the endonuclease that is responsible for removal of the intron is not in the nucleus in yeast, but it's on the outer mitochondrial membrane, which is in the cytoplasm. Okay, I said, oh my God. So I have to go back and start all over again. This was work that Anita Hopper and Yoshihisa independently discovered, so that the splicing of the yeast introns occurs in the cytoplasm and not in the nucleus. And I think that's illustrated here. So what we find in the vertebrate system is that all of these things happen in the nucleus, and then you get export. But in the yeast, splicing occurs out here on the mitochondrial membrane. And so then the question arises, is there any kind of proofreading for splicing in yeast that is equivalent to what actually I didn't show that data, but efficiency of export is much better with tRNAs that are spliced from the Xenopus system. Well, it turns out also that these two groups who were studying the cytoplasmic splicing also independently discovered that these tRNAs that are in the cytoplasm don't just remain in the cytoplasm, but amazingly, they go back into the nucleus. And there's an RNA import system that brings tRNAs from the cytoplasm into the nucleus. And this is sort of constitutive, so it's as if this tRNAs cycle into the nucleus regularly. And this RNA import then exposes the imported tRNAs to whatever is going on in the nucleus. And these tRNAs can then become aminoacylated and brought back out into the cytoplasm. Or if they are not aminoacylated, they can undergo the addition of poly A on the 3' end of these tRNAs. And the polyadenylation of the tRNA in the nucleus actually destabilizes it. And this is different from what Lynn was telling you about with messenger RNAs the other day, where she was saying that messenger RNAs become destabilized when they lose their poly A in the cytoplasm. Okay? tRNAs, when they gain an oligo A, it's not very long, but they get poly A on the end in the nucleus, it makes them a substrate for nuclease that's in the nucleus, and they become degraded. And so this is probably a monitoring system that the yeast cell uses to monitor the tRNA, the integrity of the tRNA inside the nucleus. Okay. So what I've shown you is that the nucleus versus cytoplasmic compartmentalization of the cell is very important. You need to take things from one side of this envelope into the other and back in again, and it allows for a good way for the cell to monitor what is ready to function and what can work. And so I just want to leave you with a message. It's important to integrate both biochemistry and cell biology. And with that, I want to thank Elspeth Lund, who was involved in all the experiments I showed, and the export and 5 experiments were done in collaboration with Ulrike Kutte. Thank you.